Let me introduce about MSI team, one of the research teams in Fukusaki Laboratory. MSI stands for Mass Spectrometry Imaging. This video introduces the outline of mass spectrometry imaging and research results in Fukusaki Laboratory. Seeing is believing. This is the key phrase for the MSI team. From long ago, researchers would like to see what we cannot see using new technologies. As the saying goes, seeing is believing. Visual information is intuitively understandable compared to the complicated numerical information. One of the conventional technology is microscope. Using microscope, you can see structures, but you cannot find components. Imaging mass spectrometry is one of molecular imaging technique using mass spectrometry. Using this technique, you can see many kinds of components without labeling. This is an overview of mass spectrometry imaging. Mass spectrometry imaging is a two-dimensional method of performing mass spectrometry on tissue sections. First, body tissues such as mouse brain and plants are sectioned. Molecules in the tissue are ionized by irradiating the tissue section with a laser for mass spectrometry. By repeating this on a two-dimensional tissue section, the position information and mass spectrum on each position can be obtained. Then, you can select the peak that appears at MOC of the target compounds and obtain the distribution image of that compound. Mass spectrometry images allow us to know what kind of substance, where, and how much exists from this image. This is an example of using mass spectrometry imaging on hot pepper. As you know, when you eat hot pepper, you can feel the spiciness of capsaicin, which is a pungent compound. However, it is impossible to know which part is pungent just by looking at the organizational structure of the capsicum as shown on the left photo. Therefore, we use mass spectrometry imaging to visualize the distribution of capsaicin. Then, two peaks derived from capsaicin were observed in the mass spectrum. By selecting these peaks and generating image, we found that capsaicin was accumulating in the placenta rather than the pericarp. At Fukusaki Laboratory, IM Scope Trio, manufactured by Shimazu Corporation, is used for mass spectrometry imaging. IM Scope Trio is equipped with an optical microscope for observing the sample morphology, a laser used for ionization, and a mass separator for measuring the mass of ion molecules. The spatial resolution of the generated image achieves up to 5 micrometers. It is also possible to superimpose and compare optical image and imaging results. Our research team is trying to solve many biological problems using mass spectrometry imaging. Our research target is very wide. For example, in medical field, we target metabolites of cancer tissues and neurotransmitters in mouse tissues. In food research, we do not only study on agricultural products, but also processed foods, such as rice, coffee, and also crude drugs. In addition, the metabolites from bacterial biofilm and Drosophila melanogaster are also visualized by our team. At Fukusaki Laboratory, we are working on the development of the applications and methods for a wide variety of samples. And from now, I would like to introduce recent research achievements. This is the metabolite imaging in rice koji. Koji is an important ingredient in the production of sake, and it is said that the quality of koji greatly affects the quality of sake. Generally, The quality of koji is judged by observing its structure. However, the metabolites affect koji structures is still unclear. Therefore, it is important to review the relationship between the quality of koji and its metabolite distribution. Since this study is the first time report of the analysis of koji using mass spectrometry imaging, and the challenge of this work is the size and the texture of the koji, which is very small and fragile. So, the purpose of this study is to develop the mass spectrometry imaging method for metabolite imaging in rice koji. By using cryofilm for the sectioning and supplying the matrix by sublimation, choline and the lysophosphatidyl were successfully discovered by our method. This is the research about the pesticide imaging in plants. Although pesticides is well studied as being taken up by plants and transported to all tissues of the plants, however, there are few studies on the pesticide dynamics in plants. By using mass spectrometry imaging with our developed workflow, the dynamics of ionicotinoids in tomato was successfully visualized in various parts of the plant tissue as shown in the photos. And this is another work from our team. Development of the structural isomer separation method in mass spectrometry imaging. There are multiple steroid hormone isomers in the adrenal glands. One of them is aldosterone, a compound involved in endocrine disease. In the process, to the trace amount of aldosterone, isomers are present. So, it was necessary to develop a method for detecting them separately from the isomers. Therefore, derivatization was performed using a reagent that specifically reacts with a specific structure of the steroid hormone. Then, the structural analysis with mass spectrometry was performed. As a result, we succeed in detecting aldosterone and isomers separately in adrenal gland. This year marks the 6th year of our MSI team. Many papers were published in 2020, and if you are interested, please feel free to contact us. Thank you very much.